Hi, I'm Mark. I run a company called Plasmid, which I call, it's a SaaS company, sequencing as a service. Uh, no, no software. Okay. Um, anyway, we are a company that specializes in sequencing for customers, and a lot of what we do is nanopore sequencing, and a lot of that is plasmid sequencing. So just wanted to tell everybody today about, sort of how that has happened and what a great success it's been for us, and encourage everybody to make sure this is offered at your institutions. Just a little background on our company. So Plasmid is the company, also called Primordium Labs, depending on which city you're in. We have a bunch of location or drop boxes throughout the world. We currently have about 500 drop boxes, and I know map is not entirely accurate because we have a drop box in Hawaii as well, which is not on here. But realistically, if you're in biotech hub of Boston, San Francisco, Southern California, or London, Cambridge area, we do overnight plasmid sequencing. So you'll get it probably around midnight if we pick up, say, around 5 o'clock. And then for anybody else in the US, you should have your data by the time you get in for breakfast. So less than 24 hours overnight. I'm going to give, over the course of the talk, I'm going to talk a little about our experience and how we got here. But first, I just want to tell you, why people would use us, or why people would be using plasmids, for those people who might not be familiar with that. So, for anybody who's interfacing with biological systems, DNA is the language of biological systems, and if you want to interface with them, you need to be modifying DNA and introducing it to them. And the easiest way to do that is through a plasmid, which is a small circular piece of DNA that's easy to manipulate in the lab. And then after you manipulate it, you need to verify that those manipulations are what you intended them to be. Traditionally, you would use Sanger sequencing for that. But what we found is that Nanopore is a far superior alternative. Our sort of the product categories that we think about are, pharma, so classic drug discovery, traditional biotech, so like biologics, environmental monitoring, remediation, antibody discovery, nanobody design, basic research, gene discovery, goes on and on, agriculture, synthetic biology. We even do a fair amount of stuff with like tissue culture meat, lab-grown meat companies use us a lot. And so, I'm sure everybody in here has their unique thing, but it's broadly used across the industry. So typically when people are, sequencing their plasmids, they use a technique called Sanger sequencing, which everybody's probably familiar with. As I was putting together this talk, I was trying to find a good representative picture of Sanger sequencing. So I went back to the original paper from Fred Sanger in 1977. So this is figure 1 of his paper that he got the Nobel Prize for 3 years later. And this was in December it was actually almost exactly 46 years ago that this came out. And he got the Nobel Prize, I guess, 2.5 years later after that. But this is just, if you're looking for science communication, this is just a beautiful figure, right? The entire thing is clear there. And I hope everybody here is familiar with Sanger sequencing. In the 80s, it was automated by ABI. So the traditional way that would work is you put your DNA in there with primers. It'll fluorescently label them, automatically give you the sequence trace back. And that's sort of where stuff has stayed, for 46 years. It was revolutionary when it came out, but it's still the bread and butter for sequencing. There's been sort of this cult of Sanger, which is that if you modify your DNA, modify a plasmid, you need to sequence the region that you modified, but you don't need to sequence the rest of the plasmid. And that's sort of a convenient assumption that people have been working under, but as we've learned with our customers, is entirely wrong. You need to be understanding the entire context. So I'm going to start now with sort of how we stumbled into this, and then what we've learned about that. So I was at Caltech as a postdoc working on an academic project. So this was a lineage that we were developing a lineage system where cells, as they divided, would recombine their DNA, and then you could go in and visualize them and use a phylogenetic tree analysis to figure out how related all the cells in that colony were. And as a, the way this worked is we put these recombinase sites, so Phi-C31 and BXB1 recombinase sites, and we had to put these inverted recombinase sites right adjacent to each other. So that's the @P sites on there, and they form that DNA hairpin. And what this actually meant was that these were impossible to sequence. Okay, so we tried to sequence them with Sanger, and it was actually just impossible. And so this is what our construct looked like. The barcodes are in the rainbow colors on the right-hand side of the screen. And these are our Sanger sequencing traces. And you can see that a bunch of them, in this region, they don't actually span the hairpin. And so if we zoom in on that, you can see that the sequencing traces stop right there. And so those red lines are traditional Sanger sequencing reads. So then if we zoom in on exactly what's going on there and look at the Sanger sequencing file, you can see that it just stops right at the hairpin. And no matter what we did, we could not sequence through that. And we had a really hard time wanting to publish this if we couldn't say what the sequence of the thing we were working on was. As a synthetic biology lab, it was very important to us. So I remembered my friend, Adam Rosenthal, who's now a professor at UNC Chapel Hill. He'd sent me this email, which was almost exactly 10 years ago, and said, hey, there's this Nanopore thing. It sounds like it's going to be really cool. And so my colleague in the Elowitz lab at Caltech, she'd done her PhD working on single molecule sequencing and trying to commercialize that. And I said, hey, Ding, is Nanopore a real thing? This sounds too good to be true. You just drip your DNA onto a USB drive and you get your data out? And she said, I don't think it's real. I don't think it's ever going to work. But if it did, it would be the only thing you would ever use after that. So then, a couple of years later, when we needed a new way to sequence those constructs that were impossible, this was in 2016, March or February of 2016, I think is when we first did it. The MinION Mark I was available. We ordered it and then decided to try and sequence our plasmid that was impossible to sequence via Sanger. So what we did is we cut the plasmid with a unique enzyme ICEU1, which we put in all of our backbones in case we ever needed a unique site. Ligated on the sequencing adapters, so ligated on adapters so that we could ligate on the sequencing adapters on top of that and then put it on a MinION. And we got the data back, and the data was noisy. But we got thousands of reads for each plasmid. And so it was okay, well, if we just take the average at each position, it actually looks really, really good. And it went right through the hairpins. There was no evidence that they were causing any artifacts in sequencing or anything like that. And so when we compared it to the Sanger sequencing, well, Sanger just it just died right at the hairpins, whereas this went right through. So to me, that was sort of an aha moment that, yeah, this is ultimately what's going to replace Sanger. There is no reason anybody's going to be using Sanger in the future if you're getting single molecule reads. And you can imagine your mind sort of reels at all the possibilities with that. So, I don't know if you guys have seen on Twitter, there's been a lot of people talking about ChatGPT images and how you can make it better. So I just asked ChatGPT an image of a scientist reviewing Sanger sequencing data. Okay, it's not exactly Sanger sequencing data, but it's okay. So, okay, why don't we add Nanopore data? Okay, well, not bad, it's kind of what it looks like. Okay, what if we add even more nanopores? Okay, a good start, but we need more extreme nanopores. Oh, okay, great. More nanopores. Okay, I don't know what's going on here, but maximum nanopores we get here. And so I think, maybe at NCM, 2033, we'll be putting everything in the universe through a nanopore. But, for now, it was really exciting. So we were just, at that time, and that was, 7 years ago, just really excited about the possibilities of what we could do with it. And so now I'm going to just talk about using whole plasmid sequencing, why you would want to use it, versus Sanger. So why would you use Nanopore instead of Sanger sequencing? The first thing is sort of trivial and simple, but it actually makes a big deal. That's that you don't need to use primers at all. and I'll talk about why that matters. Sanger has constraints that people just pretend like are not there when they talk about, it's the gold standard for this or that. It actually doesn't work in many cases. And so it can't sequence through hairpins. It has serious issues with high GC content DNA, and it can't sequence through highly repetitive DNA. And then, importantly, you need to get the entire picture of your plasmid. And I'll go through some customer stories in a bit about people who'd lost a serious amount of their lives working on a plasmid that they just didn't have what they thought it did in it. And I'll just say also, I will show some data later and some example constructs. In some of those cases, I've replaced the actual customer data with example data that's similar. Okay, no primers needed. So, I'll assume everybody in here has thought about Sanger to some level. And what that means is you need to design primers, right? And what that means is first you got to decide how far apart do I need to space my primers? Then you got to open up your DNA sequence, find a good site to bind that, because the primer is going to determine what region is sequenced. Okay. Then you got to log and order it. You got to pay for it. You got to wait for it to arrive. Then it comes in. You got to rehydrate it, aliquot it out, include it with your sample, and send it off. And it's sort of like, oh yeah, those are all easy things to do, but they take a serious amount of time. And when we were scaling this up at Caltech, I was running a biofab there where we were making like hundreds of plasmids every week. And dealing with that when we were doing sequencing of stuff that we'd de novo synthesized, it just did not scale. And so here's just an example of the sort of thing we had to worry about. So this is an example construct that we were working with. This is the region that we needed to sequence. Okay, so we would go in, add a bunch of primers, do Sanger sequencing, and got like most of the cover, like it mostly covered the regions we wanted. This is an example of what good Sanger sequencing data looks like. But there was like a region that was not fully covered that included the stop codon. And when you're sequencing something that you've modified in cloning, the majority of errors happen where there was either a Gibson junction or where there was a primer. And I can, having looked at whatever millions of samples, I can tell you that. So we could not get the stop codon. And so I said, okay, told my technician, you got to go back and you got to try again. So she designed, 2 more primers to try and get through this. Submit them out. Okay, they still couldn't go through that stop codon. When you zoom in, the sequence just turns to crap when you get to that stop codon, which is a fairly common failure mode with Sanger sequencing. Went ahead, designed another primer the other opposite direction, and then got good sequencing data. Okay, that's fine. It eventually did work, but just troubleshooting that simple failure case lost us about a week, right, in designing this. And the way I think about time on these experiments, and I'm sure people in this room do too, is, what are we doing? We're trying to improve, a bunch of different areas. And, if you're enabling somebody to make a discovery that's going to lead to a cure for a disease. So just, as an example, non-small cell lung cancer kills about 200 people every day, right? I think we all believe there is a future when that will be cured, right? That's what we're working toward is cures for all of these diseases, right? And if you can accelerate that by just one day, right, that's 200 people that are going to be alive because you were able to do those things. And so when we're talking about accelerating something by a week, that's a big deal, right? That has real world ramifications. Okay, getting back to what we're thinking about, we took the same plasmid, this was early on, and we just did a Nanopore library prep, okay? And then the next day we have this data. So just take an ensemble data of Nanopore, right through the whole thing, no troubleshooting. So we're like, okay, that's great. We should just do that instead of sending everything for Sanger. The other issues with Sanger sequencing are like GC-rich regions. So we were curious how well that's going to compare there. Here's real Sanger sequencing data for the PCAG promoter. PCAG is about the strongest mammalian promoter that you can use for expression, and it's extremely GC-rich. So you essentially can't sequence through it with Sanger. And so we got plasmid maps, and the person I got it from, I said, hey, I'm having trouble sequencing this region. Do you have any ideas about what I can do to get through? And he said, well, I've never sequenced it. And that was sort of the case, is like if something's hard to sequence, so you just say, oh, that's probably right. right? And a promoter region, it probably is not a huge deal if it's off, but we didn't know what the actual bases were. Um, when we put this on Nanopore, it just went right through there, no problem. And all of a sudden, the sequence that was actually in the tube was different than the sequence that we've been giving. And to this day, our customer service all the time is getting people saying, "Hey, your sequence for the PCAG is different than the map I have." And the real is like, yeah, the maps are all wrong because nobody ever can sequence it. All right, another thing we're interested in is repetitive DNA. Okay, so sequencing repetitive DNA is not fun, or at least unless you're using Nanopore. So, how well has it worked in our hands? Here's again Sanger sequencing. This is just 8 repeats. There's an 8. This is a TRE or a TET-O repeat, 8 of them. There's only 7 shown on the slide, but there's 8 in this construct. And just sequencing across that, the entire region there is it's like 300 bases or something like that. Could not span that. All of those short reads there are only like 100 bases each. It's just not possible. And so then when you start to get into higher and higher numbers of repeats, there's really just nothing you can do. And you can start to look at sizes and try and do restriction digest and figure it out. But realistically, this is an inherent problem of sequencing by synthesis, is repeats. And furthermore, if you have repeat regions that are longer than your read length, whether it's Nanopore or whether it's Sanger or next-gen sequencing, you're not going to be able to assemble that, okay? Meanwhile, we get customers who will send us things that have over 100 tandem repeats and no problem whatsoever. Nanopore goes right through it, no problem. The biggest problem is the bioinformatics on the back end of trying to align those because it breaks Minimap. But yeah, no problem, just amazing. So here's a customer story. So at Caltech, we were working with a high-profile lab, and they'd made a mouse line. They're studying the immune system, and they had this TET-inducible system. So the way this works is you feed the mouse tetracycline. That will turn on expression of the gene, and then you'll look for the change that happened after you fed them the tetracycline. It's just a common thing that happens when you're studying these. And they kept feeding the mouse the tetracycline, and nothing. It would never work. And so they thought, like, is our system wrong? What's going on? So they sent us the original plasmid that they'd used to make the mouse. And this is what their sequencing data looks like. Okay, there's a giant gap right where all those repeats are. And so we looked, they actually only had 2 repeats instead of, I think, 18 that they were looking for. And the customer, this is a high-profile lab, a very good postdoc, lost 5 months essentially of making and characterizing this mouse strain and trying to figure out why it wasn't working. Whereas if they'd just done the sequencing on the front end, it would have eliminated that. And you might say, well, they should have, if you're going to do that, you should just sequence it by any means necessary. Well, the problem is that you can't sequence that with Sanger. So the way these things would typically work, when you have difficult sequence regions, what'll happen is somebody will characterize it once in history. Maybe they'll do a restriction digest or something like that. And then everybody will just assume it's right forever because nobody wants to go back and redo that. But the problem is that the repetitive DNA, especially if the hairpin's in it, is extremely unstable in E. coli. And so those will just pop out rapidly. And so now, they send us every construct, and most people do. Yeah, another thing, when you're going to design primers, you have to decide how far apart am I going to space them. And so, say a good Sanger read is going to be 1,000 base pairs. A bad read would be, 500, 400. So you say, okay, do I want to space them every 500? That's kind of excessive. It's a lot of work, or maybe I space them out. So it's just like this mental burden of figuring out how to space them. Because if you go too short, then it's more work. If you go too long, you're going to be in this situation where, you have your primers spaced apart. You don't quite get through the region, the whole region. And then what do? You just say either what you do is you go back and spend another couple days sequencing this region, or you're like, that's probably fine. It's just a little window that I couldn't sequence. And of course, that always comes back to bite people. But even in this simple case, that project is delayed by 2 days just because they didn't get sequencing coverage. This next one is somebody would contacted our customer service to tell us she was a grad student, had received a plasmid from the previous grad student in the lab, and could never get the system to work. And her PI thought she was an idiot because she couldn't get anything to work and whatever. Eventually, after over a year of troubleshooting, she sent the plasmid to us for sequencing, realized that it was missing some important stuff on the other half of the plasmid, and that she essentially just wasted a year of her life trying to hunt that down. And it's like for $15 could have, totally eliminated that. Something else we're learning a lot from nanopore sequencing. So one of the crazy things about nanopore, right, is you're getting a single molecule read from one end to the other, right? And if you can do your preps cleanly, you'll get the entire plasmid sequence. And what that means is that you can discriminate between a monomer plasmid and a dimer plasmid. And you cannot do that with Sanger sequencing. You can't do that with Illumina sequencing. But this happens all the time. It's actually not generally a huge issue when it does happen, but it does happen. And it happens, I would say, probably 5% of the time or something like that. This is the plasmid that somebody ends up with, so like 1 in 20 times. And it's, if you do your ligations at a very high concentration, or you do your Gibson assembly at a high concentration, this happens. So here's this is the picture that's derived from this one. So the customer was expecting their plasmid to be at about 5 kb, but they observed it about twice as big. And when you look at the gel, you can see that essentially all of or you look at the histogram, essentially all of the reads are the dimer. And this is just, I guess I should be describing this a little bit better. We provide our customers with a virtual gel because that's what molecular biologists are used to thinking about. That's essentially just taking that histogram and converting it into a heat map. If you're working with industrial strains, which are typically Rec A positive because they grow a little bit faster and a little bit more robust, they tend to multimerize just naturally. So this plasmid probably started out as a monomer, but just as it was growing in that industrial strain, started multimerizing. And so sometimes when you sequence them, you'll see like an entire ladder of sizes. So at Plasmidsaurus, we have a sort of fun relationship with our customers where we ask them to draw a picture of a dinosaur on the back of their order sheet when they sent it in. And so people will all send us fun stuff like this guy. This person drew this dinosaur and says, "Are these the plasmids I think they are?" And the answer a lot of times is no. So we've got, we know people have ordered plasmids to be synthesized from professional companies. They come back and they got the wrong plasmid, right? Somebody picked it out of the wrong tube. I won't name any names, but that does happen with many of the places people order from. Another thing that happens in your tube is you get deletions. And so here's an example of, this is the histogram of the read lengths. And so you can see there's a lot of them at 10.5 kb, but there's another peak at about 3 kb. And when you look at it, this is an IGV view, there's a deletion, essentially a 7.5 kb deletion in their plasmid. And this is actually very common, and people have known about this historically, is that if you're growing lentiviral vectors, you need to try and grow them in stable cells because they've got these repeats that'll just pop out your insert. So here's the parental vector. This is the 10.5 kb one. And then here is one where the shorter vector, and you can obviously, we have the algorithms that'll go in and give you the sequences of those different peaks. And essentially what happens is the two LTRs on this lentiviral backbone just recombine and pop out everything in the middle. And this is also, surprisingly, invisible on Sanger sequencing. So you will get customers that send us a tube. We'll tell them the majority of the stuff in their tube is the smaller thing. And they'll say, "No, I Sanger sequenced it. Like, you don't understand. It's already verified by the gold standard that this has the insert in there." And what happens is if you have 90% of the stuff on the bottom and 10% of the stuff on the top, but your primer is specific for the thing on the top, you're going to get good quality sequencing data for the thing on the top, even though the vast majority of what your tube is not that. And so then you'll go into your prep, and you'll wonder why you've got no viruses or why everything's messed up. And that just happens like all the time, like every day we see that. Another thing you get with Nanopore is you get discrimination of mixtures of similar plasmids. So we also provide these AB1 files to our customers. And so these are artificially generated from the underlying Nanopore data because just people are used to looking at the AB1 chromatograms. And so this customer had a region that looked kind of weird like this. So when we go in and look at the underlying IGV views, you can see there's actually a mixture of two plasmids there that you can deconvolve and give to the customer. Another thing that happens is you might have received something like this. Maybe some of the older people in the audience have seen these before. This is what plasmid masks used to look like before there was good DNA editing tools. And you would get this, and you would say, okay, I know what restriction site I can clone into, but you don't know anything else about the plasmid. So when we sequence on Nanopore, what we'll get is a string, right? That's the entire length of the plasmid. So it'll be like 10 kb of just DNA. And then our lead bioinformatician, Matt McGuffey, who's here, he developed a tool called Planotate. And what Planotate will do is it'll take your plasmid sequence, it'll search all of the feature databases, pull the features into that plasmid, generate an HTML, interactive HTML map, as well as an annotated GenBank file that we can then return back to the customers. So they'll see exactly what's on their plasmid. And that's an open source tool that anybody can use. So yeah, it's used. I think anybody who does whole plasmid sequence is actually using that tool. So yeah, Matt's over in the bioinformatics session, but say hi if you see him. And then I just want to go over a couple other things before I finish up here. So we do not just plasmid sequencing on Nanopore, we also do PCR sequencing. We do plasmids isolated from colonies, and we do bacterial and yeast genome sequencing. So for PCR sequencing, this is just the NEB ladder. So ladder right from neb.com, New England Biolabs, and sequence it. And if you were to sequence that, with another method, you just get a mixture of everything, and you can't default. Here we can go in and we can say, okay, what is the sequence at 8 kb? What is the sequence at 6 kb? Totally easy to do based on the sequencing technique. Another thing we do, so sequencing directly from bacterial colonies. These are some cool plasmids we got from Sebastian Kosioba, who runs Binomica Labs. These are just fun colored plasmids, but we like to use them as our controls for when we're growing stuff because they're just fun to look at. And then the other one, customer, I told you they make dino art for us. This person actually made it out of fluorescent bacteria, which is kind of fun. But we can receive, bacterial colonies into our lab, do that prep, same day prep or same night prep, and then extract the DNA and run it on Nanopore and get high quality DNA to the customers same night. Another thing we do is bacterial and yeast sequencing. So again, we get those samples in the morning, get them on the sequencer in the afternoon, and then customers will get their data 24 hours, which has been a game changer for people that are doing microbial genome engineering, for instance. I don't know if I want to go through this. This was just, we have, our Twitter page is just full of lots of people who are sort of very happy. There's literally like 20 slides like this of people that are super happy. But I think I will sort of wind it down there and then just also say, I know a lot of people here are from Core Labs. So we do partner with Core Labs. Like I said, we have drop boxes all over the country. We probably have one very close to you. And we can work with you guys to, get overnight sequencing if you want to reach out. So you can find me or Kevin Coe, who's back there, or email us at support@plasmidsaurus if you are interested in doing that. And I think I'll leave you with this. So my son made this animation for us. I'm gonna leave you with it. Okay, thanks.